Chapter 3. Stereotype Behavior. The stereotype is a French word derived from the Greek words sigma tau epsilon rho epsilon, stereos, and tau pi omicron, typos, which mean a solid impression on one or more idea/theory. The term comes from the printing trade and was first adopted in 1798 by Firmin Didot to describe a typography-duplicated printing plate. Typography is the work of arranging and setting types and then printing from them. The first reference to stereotype was in 1850 as an image perpetuated without change. It was first used in modern psychology in 1922 as a public opinion. The generalizations are used in North America as the perceived stereotypical behavior. To characterize or regard as a stereotype is to give a fixed form to a person, place, or thing. In social psychology, a stereotype refers to an overgeneralized belief about a particular category of people. Generally, on any given level, people's perceptions are influenced by their expectations. Stereotypes start in childhood, at church, home, school, and especially with neighbors. Stereotypes can be positive, negative, or neutral. Stereotypes can be beliefs about people based on their membership in a particular group. Ethnic stereotypes are widespread and shared by members of social groups. Stereotypes exist in different cultures, ethnic groups, and subraces. Stereotypes based on ethnicity, gender, or occupation are common in many societies. Psychologists believe that it is a natural aspect of human behavior and that it is beneficial to each group to identify with one's ethnic group. Racial stereotypes. A famous study on racial stereotypes was published in 1933 by Brawley and Katz. The reported questionnaire was completed by students at Princeton University in the USA. Students there had clear but negative stereotype views. These were white Americans at the time, and other ethnic groups in America still were embodying slavery. Their focus on other ethnic groups included Americans as industrious and intelligent, Druzes as mercenary and shrewd, Japanese as shrewd and sly, and Negroes as happy-go-lucky, ignorant, lazy, and musical. They viewed the white subrace as ambitious, industrious, and progressive. Later studies conducted in 1951 and 1967 were more positive, but ethnic groups still held critical views of other ethnic groups. Since then, other ethnic groups in America have been able to achieve a better quality of life without being enslaved. When comparing discrimination to prejudice concepts, discrimination is a behavioral component of the cognitive mental processes, whereas prejudice is the affective component of stereotyping. Verbalizing a prejudice can be perceived as expressing ill-founded ignorance or prejudices with a need for justification. People who express explicit stereotypes tend to express the unconsciousness of individuals who appear to have no awareness or control over their ethical norms. They may contain conceptualization theories and contradictory elements and can be used as forms of discipline. Explicit stereotypes are unwilling to rethink one's attitude or behavior. Many people argue that racial stereotypes are a way to justify racist attitudes and behavior. Prejudice tends to legitimize discrimination. It justifies one group's dominance over another. Prejudice can boost the self-esteem of stereotyping people and lower the self-esteem of people on the receiving end. Either way, both the person on the receiving end and the person doing the prejudging generally have behavior issues that need to be worked through. The overall perspective of racial stereotypes: it is very common to overgeneralize either by belittling other subraces or favoring your own, and every ethnic group has racial stereotypes of other groups. According to the contact hypothesis, prejudice declines when in-groupies become more familiar with the attitudes, customs, food, music, and norms of people of an outgroup. Contact with the outgroupie helps people to see the diversity among its members. Hostility between an in-groupie and an outgroupie increases when the groups compete. Researchers have found that hostility between the groups decreases when those groups must cooperate to reach a shared goal. In such situations, people in the two groups tend to feel they belong to one larger group rather than two separate groups. Research shows that conflict and prejudice among groups can be reduced if four conditions are met. The groups have economic opportunity, equality in terms of legal status, and political power to advocate equal rights. Also, the groups have opportunities to interact formally and informally with each other, while the groups cooperate to reach a common goal. Image versus reputation. An image is a physical likeness or representation of an animal, person, or thing, referencing something such as a painting, photograph, sculpture, or something otherwise made visible. A mental representation of an appearance, company, conception, embodiment, figure, formation, idea, idol, illusion, notion, reflection, type, etc. In psychology, it is a mental representation of something previously perceived in the absence of the original stimulus. General reputation is the estimation in which a person or thing is held, especially by the community or public, of which it is possible to achieve a favorable reputation or good name that isn't ruined by a reputation of misconduct. The image or reputation of a social entity refers to an organization, a person, or a social group, refers to an opinion about that entity, generally because of evaluation according to the socially set criteria. Reputation is a highly dynamic phenomenon in two distinct senses. It is subject to change, especially as a result of corruption, deception, errors, etc. It emerges as an effect of a multilevel, bidirectional process. Reputation is an efficient mechanism of social control in natural societies. It is a subject of study in management, social, and technological sciences. Research has found it takes approximately 3.5 years to fully recover reputation. The influence extends to competitive settings such as communities, firms, groups, organizations, and institutions. Reputation acts on various levels of agency, individually and superindividually. On the superindividual level, it interests collectives, communities, groups, and abstract social entities such as civilizations, corporations, countries, cultures, firms, and organizations. It affects the circumstances of everyday life to relationships between nations. Reputation is a fundamental instrument of social order, based upon spontaneous social control and its distribution. While the concept of reputation is considered a component of identity, it is important in business, communities, education, and other fields. Reputability is rooted in being an honorable and respectable person, of which involve consistently being a law-abiding citizen and upholding standards. Discontentment with the responsibilities of being a law-abiding citizen is rooted in distrust and a lack of embracing the national flaws and self-glory. While it does take courage to stand for something that is rooted in aspects of being a citizen, it doesn't take courage to complain. Compulsive behaviors. An addiction is a state of being enslaved to a habit or practice. Something that is psychologically or physically habit-forming, such as alcohol or narcotics, to the extent that its cessation causes severe trauma. Compulsive personality is governed by an obsessive need to conform, be scrupulous, etc., coupled with an inability to express positive emotions. It is a character that pertains to compulsion. Obsessiveness causes an obsession. Someone who obsesses behaves obsessively, and the person excessively or extremely does so. Compulsive behavior is performing an act persistently and repetitively without it necessarily leading to an actual pleasure or reward. Compulsive behaviors are viewed as a need to reduce anticipation caused by internal feelings, of which the person wants to abstain from or control things perceived as adversity, misfortune, suspicion, or fear of future evil or trouble. Whether this is an idea, opinion, or view on a subject. Compulsive behaviors can be extremely biting nails or skin to the point of obsessive-compulsive disorder, OCD. These are self-mutilating behaviors. Also, compulsive behaviors include excessively checking things, counting, gambling, hoarding, overeating, repeating, sex, shopping, stealing, talking, trichotillomania, washing, etc. Addiction is a compulsion toward a rewarding stimulus. An addiction could be linked to the beliefs, concepts, customs, and practices of a person's upbringing. Compulsive behavior is the core feature of compulsive disorder, OCD. Compulsive behaviors could be linked to forgetfulness or neglectfulness. The act isn't disturbing pathologically when it is a repetitive, small, restricted behavior. Compulsive behaviors. Checking, repeatedly checking appliances, door locks, or light switches for possible danger or harm. Counting, repeatedly counting specific numbers of importance relating to a situation, and when the number is considered significant, the person performs more duties. Eating, repeated overeating of foods low in nutrition is the inability to control the amount of nutritional intake, resulting in excessive weight gain. Overeating generally becomes a coping mechanism for dealing with stress. This behavior usually develops in early childhood. The feelings are accompanied by guilt and shame of using food to avoid emotional stress. Other issues can be binge eating, depression, and withdrawal. Gambling, repeatedly gambling assets or money away and not resisting the desire to gamble. This can lead to personal and social issues for an individual. This behavior begins in early adolescence for men and women between the ages of 20 to 40. Other issues can be feuds with family members, others, the law, and avoiding stressful times. Two major issues revolve around a lack of money to keep gambling or paying off debt. Hoarding, repeatedly saving possessions, believing those items will be useful in the future or have sentimental value. Other reasons can be afraid of losing documents, information, and object characteristics. Usually, the items that are saved include books, clothes, containers, craft items, junk mail, and newspapers. Other issues can be blocked exits that can pose danger to family and friends. Occurrence, repeatedly dismissing or ignoring accusations of having obsessive-compulsive disorder. Researchers say about 50 million people are suffering from some type of OCD. Sufferers appear more secretive than other people with psychological problems and may endure the condition for years before seeking help. Sex, repeatedly behaving and thinking about sex-related desires, including feelings that are considered illegal and or culturally and morally unacceptable. This disorder is also known as hypersexual disorder, hypersexuality, nymphomania, or sexual addiction. Shopping, repeatedly buying and shopping, believing that you ought to be a trendsetter or the first to purchase new products. Other issues can be that shopping causes impairment in a person's life with family or finances. Approximately 80% more women suffer from this type of behavior than men worldwide. Yet still, there is no proven treatment for this type of compulsive behavior. Stealing, repeatedly stealing things, believing you are to achieve success by any means necessary. This can lead to identity, personal, and social issues. Other issues are a bad reputation, run-ins with law enforcement, incarceration, or even the death penalty due to an innocent bystander. Talking, repeatedly beyond the bounds of what is a socially acceptable amount of talking. Not leaving space for the other person to give details, feedback, or an opinion. Talking about things that don't have a significant meaning or a well-thought-out plan. Other issues those actions can lead to include personal and social forms of confrontation or even obstruction with law enforcement, etc. Personality traits that have been positively linked to this compulsion include the ability to communicate, assertiveness, self-perceived communication competence, and neuroticism. Trichotillomania, repeatedly picking hair or skin of the body, which results in bald spots. Other issues may be digging, rubbing, or scratching the skin, which is to rid blemishes of the skin. All compulsions can leave abrasions and irritation on the skin and can lead to an infection. These acts usually occur in times of high anxiety, boredom, or stress. Washing, repeatedly washing things for fear of contamination, such as clothes, body, dishes, hands, etc., throughout the day. Either can be perceived as a part of a ritual that follows a pattern. External and internal stimuli. Human senses include hearing, smell, vision, taste, touch, and several others. A stimulus, plural, stimuli, is something that incites action, exertion, or quickens action, feeling, thought, etc. In psychology, a stimulus is something that excites an organism or part of functional activity. A detectable change in the external or internal environment. When an organ or organism responds to external stimuli, it is called sensitivity. A stimulus applied to a sensory receptor elicits or influences a reflex via stimulus transduction. Sensory receptors receive information, signals, from inside the body as well as from outside the body. Senses or sensors detect energy from the world and convert the energy into signals. The signal from a sensor may be high, very complex, or low, simple, frequencies. The organism processes or interprets the signal from the sensor as ignorable, positive, or negative. Positive signals mean the person is either attracted to the stimulus or wants more. Negative signals mean the person wants to avoid the stimulus. Signals may be processed near the sensor, in the brain, or somewhere in between during any given situation. Homeostatic imbalance forces a change in the body, and the receptors and sensors monitor different parts of the body. The first component of a homeostatic control system is often an internal stimulus. Chemoreceptors, mechanoreceptors, and thermoreceptors respond to chemical or temperature change in pressure or stretching. Mechanoreceptors, such as baroreceptors, detect a change in blood pressure. Merkel's discs detect pressure and touch that are sustained, and hair cells detect sound stimuli. Homeostatic imbalances can serve as internal stimuli, such as nutrient and ion levels in the blood, and oxygen and water levels. Deviations from the homeostatic ideal can generate homeostatic emotions such as fatigue, pain, or thirst. Motivated behavior then restores the body to stasis, such as drinking, resting, or withdrawal. External stimuli can produce systemic responses throughout the body. This is called the fight-or-flight response. A stimulus is detected as a high probability when its level exceeds the absolute threshold. If a signal does reach, the information is transmitted to the central nervous system, CNS. Then it is integrated to decide how to react. The CNS determines whether a signal causes a reaction or not, and commonly, why stimuli cause the body to respond. Senses. Smell. The olfactory organs consist of epithelium and lamina propria. They are located on both sides of the nasal septum. The olfactory epithelium contains cell receptors that cover the interior surface of the cribriform plate, the perpendicular plate, or the nasal conchus. Roughly 2% of airborne compounds inhaled are carried to the olfactory organs as the air is inhaled. The olfactory receptors that extend past the epithelial surface provide a base for many cilia lying in the mucus. Odorant-binding proteins interact with the cilia-stimulating receptors. The absolute threshold for smell is the minimum amount needed to elicit a response from receptors in the nose. Sound. Changes in pressure caused by sound reaching the external ear resonate in the tympanic membrane and articulate with the auditory ossicles or bones of the middle ear. The tiny bones multiply pressure fluctuations as they pass disturbance into the cochlea, a spiral-shaped bony structure within the inner ear. Hair cells in the cochlear duct, specifically the organ of Corti, are deflected as waves of fluid and membrane motion travel through the chambers of the cochlea. Bipolar sensory neurons located in the center of the cochlea monitor information from cell receptors and pass it on to the brainstem via the cochlear branch of cranial nerve 8. Sound information is processed in the temporal lobe of the CNS, specifically in the primary auditory cortex. The absolute threshold for sound is the minimum needed to elicit a response from ear receptors. Taste. Gustatory cells are located on the surface of the tongue and the adjacent portions of the pharynx and larynx. The gustatory cells form on taste buds, and specialized epithelial cells are turned over every 10 days. Each cell protrudes through microvilli called taste hairs, through taste pores, and into the oral cavity. Dissolved chemicals interact with these cell receptors, and different tastes bind to specific receptors. Salt and sour receptors are chemically gated ion channels that depolarize the cell. Sweet, bitter, and umami receptors are called gust-dusin. They are specialized G-protein-coupled receptors. Both divisions of cell receptors release neurotransmitters to afferent fibers, causing action potential firing. The absolute threshold for taste is the minimum needed to elicit a response from receptors in the mouth. Touch. Feeling is recorded by sensory receptors on the skin and then travels to the central nervous system. It then integrates a decision of response to be made. If a possible response can be made, a signal is sent down to a muscle that acts appropriately according to the stimulus. Vision, light, such as information or stimuli, enters the retina. It then excites a neuron called a photoreceptor cell. A local graded potential begins in the photoreceptor. It then excites the cell for an impulse to be passed along through a track of neurons to the central nervous system. A firm memory of smells. Memory and odor are linked due to their close physical proximity in the brain. The dorsolateral prefrontal cortex stores most memories, and memories are retrieved using the prefrontal cortex, a region located near the olfactory processing center. These two right hemisphere centers are proximal to the amygdala-hippocampal complex. The nearness of the centers for memory storage, odor processing, and memory recall explains the influence association between memories and smells. Other neutral memories, such as autobiographical memories, can be recalled frequently with great clarity and detail. The primitive forms of behavioral reactions to predatory odors are avoidance, fear, and stress. Blind sight is the ability of a blind person to sense a light source or other visual stimulus accurately even though unable to see it consciously. It is a demonstration of cognitive neuroscientists use to better explain sensory messages traveling through the primary visual cortex. The blind sight sensory messages move from the retina to the subcortical region of the brain rather than traveling through the primary visual cortex. The superior colliculus is also located in the subcortical region, which processes eye movements and performs other vision-related functions. While relying on a subconscious visual pathway, blind sight kicks in when the visual cortex or other key areas involved in vision are damaged. The eyes and optic nerves remain intact and are said to still be capable of gathering and sending information to unaffected parts of the visual system. It is believed by cognitive neuroscientists that animals and humans can embed just about any smell. But many innately aversive and predatory odors you can train an animal to consider attractive. Context plays a role in smell-triggered memories. The way you identify and process these odors is usually insignificant because you don't notice those smells suddenly. In general, you link smells to experiences that count and then savor those memories to survive. Emotional events tend to be the most vivid autobiographical memories. Over time, protective behavior patterns are reinforced through death and life situations that are genetically embedded in the amygdala. This memory retention is preserved through human evolution, for linking smells to threatening situations that could ruin survival. The flight response is the result of eons of learning how best to survive. These findings suggest that you cannot learn to change your behavioral response to particular smells. Systematic responses. In the nervous system, external and internal stimuli can elicit two categories of responses: an excitatory, inhibitory, and an action potential, which is normal. When a neuron is stimulated by an excitatory impulse, neuronal dendrites are bound by neurotransmitters that cause cells to become permeable to a specific type of ion. Essentially, the type of neurotransmitter determines which ion the neurotransmitter will become permeable to. An excitatory response is generated in excitatory postsynaptic potentials, which are caused by an excitatory neurotransmitter. Normally, glutamate binds to neurons' dendrites, causing an influx of sodium ions through channels that are located near the binding site. This membrane permeability change in the dendrites is known as a local graded potential. This causes membrane voltage to change from negative resting potential to a more positive voltage, a process known as depolarization. Sodium channels opening nearby other opening sodium channels allow a change in permeability to spread from dendrites to the cell body. A strong enough graded potential or several graded potentials occurring at the fastest frequency enable depolarization to spread across the cell body to the axon hillock. An action potential can be generated and propagated down a neuron's axon from the axon hillock, causing sodium ion channels in the axon to open as the impulse travels. Once a signal begins to travel down an axon, the membrane potential has already passed the threshold, and it cannot be stopped. This phenomenon is known as an all-or-nothing response. Groups of sodium channels opened by membrane potential strengthen the change in the signal as it travels away from the axon hillock, allowing it to lengthen the axon. As depolarization reaches the end of an axon or axon terminal, the neuron end becomes permeable to calcium ions that enter the cell via calcium ion channels. The release of neurotransmitters is caused by calcium stored in synaptic vesicles, which enter the synapse between two neurons known as the presynaptic and postsynaptic neurotic. If a signal from the presynaptic neuron is excitatory, it will cause a release of an excitatory neurotransmitter, causing a similar response in the postsynaptic neuron. These neurons communicate with thousands of other receptors and target cells through complex, extensive dendritic networks. Communication between receptors in this fashion enables discrimination and a more explicit interpretation of external stimuli. In their frequency, these localized graded potentials trigger action potentials that communicate along nerve axons arriving in specific areas of the brain. In these highly specialized parts of the brain, signals are coordinated with others to trigger a new response. Normally, if a signal from the presynaptic neuron is inhibitory, neurotransmitters, GABA will be released into the synapse. This neurotransmitter causes an inhibitory postsynaptic potential in the postsynaptic neuron. Essentially, this response causes the postsynaptic neuron to become permeable to chloride ions, making the membrane potential of the cell negative, basically a negative membrane potential, making it more difficult for the cell to fire an action potential and prevents any signal from being passed through the neuron. A neuron can be either excitatory or inhibitory, depending on the type of stimulus, though receptors and stimuli vary. Most extrinsic stimuli first generate localized graded potentials in neurons associated with the specific sensory organ or tissue. Behavior response. Russian physiologist Ivan Pavlov, 1849–1936, was a practical scientist who experimented with training animals to respond to a particular stimulus, even when that stimulus was removed. This is known as classical conditioning, a scientific concept of conditioning or learning by association with particular stimuli. It calls for pairing a stimulus that elicits a specific response with one that does not until the second stimulus elicits a response like the first. Classical conditioning is contrasted with operant conditioning, which involves administering or withholding reinforcements, rewards, based on the performance of a targeted response. Many experiments of this type demonstrated that an innate behavior can be modified. Genomic imprinting. While a genome is a full set of chromosomes, they are all the inheritable traits of an organism. An imprint is a chemical mark made by pressure, essentially a figure or mark impressed or printed on something. In the psychology of animal behavior, imprinting is rapid learning that occurs during a brief receptive period. Typically, soon after birth or hatching, it establishes a long-lasting behavioral response to a specific individual or object, as attached to an offspring, parent, or site. Genomic imprinting is epigenesis that results in monoallelic gene expression according to a parental origin, whereas genes are expressed in a specific manner according to the parent of origin. Independent of classical Mendelian inheritance, genomic imprinting is an inherited process. The epigenetic process involves DNA and histone methylations without altering the genetic sequence. These epigenetic marks of the parents are established, basically, imprinted, in the germline, egg cells or sperm, and are maintained through mitotic cell divisions in the somatic cells of an organism. There is a lack of time for reprogramming to be completely achieved during somatic cell nuclear transfer. Imprinting may cause problems in cloning, with clones having DNA that isn't methylated in the correct positions. The nucleus is added to an egg during somatic cell nuclear transfer, and then the egg quickly starts dividing within minutes after that. Unlike the days or months it takes for reprogramming during embryonic development. If time is the factor, to give time for proper reprogramming to occur, it may be possible to delay cell division in clones. For normal development, appropriate imprinting of certain genes is important. Imprinted genes have major effects on development and placental biology before birth. There is evidence that suggests that imprinted genes also have important roles after birth. In studies, they have been shown to exert important effects on postnatal development, growth, and survival on adult phenotypes. Imprinted genes are emerging as key regulators of metabolic processes, influencing the maintenance of body temperature, food intake, and adiposity by acting on multiple pathways and tissues. Many imprinted genes are expressed in the brain and affect diverse aspects of behavior, from adult social behavior to infant feeding and sleep. Cognitive neuroscientists have found that genomic gene expression does dramatically influence brain behavior and development and may also contribute to a variety of brain disorders. Recent work on postnatal stages has shown that imprinted genes influence a wide-ranging array of biological processes, the effects of which extend into adulthood. The imprinting inherited chemical mark on a gene shuts off but doesn't delete it. Often, that mark will shut off the mother's gene, and other times the father's gene. From generation to generation, which gene is turned off or on appears to be consistent, and it is always inherited from the same parent. Both origin genes of the father and mother exert different pattern expressions in developmental shifts. The maternal genes play a 60% leading role in early development, and the paternal genes play a 70% role in stepping on stage during adulthood. While scientists do have some ideas about which parent's gene is activated during imprinting, they aren't clear on what the genomic imprinting function is, why it takes place, nor why maternal bias shifts to paternal bias either. Demonstrations of genomic imprinting have been done on animals, fungi, and plants. Imprinting was predominantly recognized in mammals about 30 years ago from embryological and genetic studies. This imprinting has evolved as a mechanism that balances parental resource allocations to offspring. As a result, epigenetic and genetic disruptions alter the specific dosage of imprinting genes and can lead to developmental abnormalities that are often associated with fetal growth and neurological behavior. Disrupted expression of imprinted genes is an important cause of human disease. Investigations of mouse mutants have been important in unraveling the roles of imprinted genes and in elucidating some of the pathophysiological mechanisms involved in human imprinted syndromes. There were about 150 imprinted genes found in mice and half that number in humans as of 2014. Imprinted genes are a contributory factor in a wide range of common diseases, such as cancer, diabetes mellitus, intrauterine growth restriction, obesity, and psychiatric disorders. Over the past 20 years since the first gene was discovered, other mechanisms have been implicated in this regulatory mode of gene expression. Epigenetic and genetic causes include at least 8 imprinting human disorders, some of which are Angelman, Beckwith-Weidman (BWS), Prader-Willi, and Silver-Russell (SRS) syndromes. Other conditions that involve imprinting include pseudohypoparathyroidism and transient neonatal diabetes mellitus. Unbalanced imprinting may be a cause of autism and psychosis, and essentially, it is an imprinting brain theory. Both Angelman and Prader-Willi syndromes were the first imprinted genetic disorders found in humans, and these syndromes are associated with loss of the chromosomal region. This region contains the maternally expressed gene UBE3A and the paternally expressed genes NDN and SNRPN. The maternal inheritance of the deletion is associated with Angelman syndrome, and it is characterized by epilepsy, tremors, and an everlasting facial expression of a smile. The paternal inheritance of a deletion of the region is associated with Prader-Willi syndrome, and it is characterized by hypotonia, hypogonadism, and obesity. DIRAS3, known as ARH1 or NOEY2, is a maternally imprinted and paternally expressed gene that is located on chromosome 1 in humans. Deletion or reduction of DIRAS3 expression is linked to an increased risk of breast and ovarian cancers. The protein encoded by DIRAS3 is not expressed in 41% of breast and ovarian cancers, and this suggests that it functions as a tumor suppressor gene. As a result, if uniparental dismay occurs and the person inherits both chromosomes from the mother, since the gene will not be expressed, the individual is at a greater risk for developing both breast and ovarian cancers. Violent behavior genes. In a developed country such as Finland, scientific research shows there are 2 genes associated with violent behavior, monoamine oxidase A, MAOA, and a variant of cadherin 13, CDH13. Unfortunately, they believe the genes couldn't be used to screen criminals as a crime prevention measure and shouldn't influence convictions. It appears they were in the early stages of research with these genotypes. However, they say that at least 5 to 10% of all violent crimes could be attributed to individuals with these genotypes. Offenders with both genes are 13 times more likely to have a history of repeat violent behavior. Criminals who commit at least one murder are believed to carry a low-activity version of the MAOA gene, unlike criminals who commit multiple murders. The low-activity version of MAOA is dubbed the warrior gene due to its link to aggressive behavior. Monoamine oxidase A, known as MAOA, it is an enzyme in humans that is encoded by the MAOA gene. The MAOA gene is one of 2 neighboring family member genes that encode mitochondrial enzymes that catalyze the oxidative deamination of amines, such as dopamine, norepinephrine, and serotonin. A mutation of this gene results in the Brunner syndrome. This gene has also been associated with various other psychiatric disorders, including antisocial disorder. In humans, the promoter of the MAOA gene is located on the X chromosome. There is a 30-base repeated sequence in the promoter region of MAOA. There are 2R, 3R, 3.5R, 4R, and 5R variants of the repeat sequence. The frequency distribution of variants of the MAOA gene between ethnic groups varies. MAOA is a key regulator of normal brain function. MAOA's function affects the following systems. 1. The dopamine system is involved in arousal, mood, motivation, reward, and other behaviors. 2. Epinephrine/norepinephrine system, which facilitates autonomic nervous system activity and the fight-or-flight reactions. 3. Serotonin system, which is involved in impulse control while affecting appetite, regulation, and sleep. DAT1 gene, known as the dopamine transporter or SLC6A3, it is a membrane-spanning protein that pumps the neurotransmitter dopamine out of the synaptic cleft back into the cytosol. In the cytosol, other transporters sequester the dopamine into vesicles for storage and later release. Dopamine reuptake via DAT provides the primary mechanism through which dopamine is cleared from synapses. Evidence points to a possibly larger role of the norepinephrine transporter, and there may be an exception in the prefrontal cortex. DAT is implicated in several dopamine-related disorders, including alcoholism, attention deficit hyperactivity disorder, bipolar disorders, and clinical depression. Evidence for the associations between DAT and dopamine-related disorders has come from a type of genetic polymorphism, known as a VNTR, in the DAT gene DAT1, which influences the amount of protein expressed. In humans, the gene that encodes the DAT protein is located on human chromosome 5, which consists of 15 coding exons and is roughly 64 kbp long. T-cadherin, known as cadherin 13 or H-cadherin, heart, CDH13, is a member of the cadherin superfamily and, due to the lack of cytoplasmic and transmembrane domains, is anchored to cell's membrane through the GPI anchor. Classical cadherins are for cell-to-cell contact, regulation of morphogenetic processes in embryos, and tissue integrity in the adult organism. Cadherins function as membrane receptors mediating inside and outside signals, thus activating small GT-pathways and the beta-catenin/WNT pathway, resulting in changes in the phenotype and cytoskeleton reorganization. Since T-cadherin is a GPI-anchored member of the cadherin superfamily and doesn't have the intracellular domains, its involvement is in low-density lipoproteins, LDL, harmonic effects on Ca2+, mobilization and increased cell migration along with phenotype changes. The exact signaling adapter and partner proteins for T-cadherin remain to be elucidated. In humans, the T-cadherin gene is located on the 16th chromosome. Advances in imaging and neurochemistry technology do show that many imaging and emotional disorders, such as anxiety, depression, suicide, and violence, involve disruptions in the brain's normal activity due to altered chemical imbalances, environmental factors, and gene expression. The violence in our society has motivated biomedical researchers, psychologists, and sociologists to look for genetic causes, markers, and predictors for such destructive human behavior. The overall variance in aggressive behavior seems to be explainable by genetic influences. Nature and nurture are genotypes included in the environmental factors that we are exposed to throughout life, and either contribute to reshaping or shaping of brain functions. Specific gene expression of the brain, such as DAT1, DRS2, and MAOA, can affect neurotransmitter levels, in turn influencing abuse, diet, sleep quality, social relationships, stress, and substance abuse. During the early 1990s, researchers linked increased frequencies of antisocial behavior with low levels of MAOA, especially when individuals had a history of being mistreated during childhood. A later investigation conducted in 2008 by Guo and colleagues revealed MAOA variants in 2,500 American boys from the 7th to the 12th grade. They demonstrated a genetic basis for severe aggressive behavior that was seen at school. The MAOA gene VNTR2R-MAOA was a risk factor for violent delinquency when the boys suffered other stresses such as failing school, family issues, or low popularity. Such studies suggest that when subject to an abusive childhood, individuals with low MAOA expression are at an increased risk of developing antisocial personality disorder, and a lifetime of committing violent crimes, exploiting, manipulating, and violating the rights of others may be the result.